this computer. Well, I think that should be recording now. And here's the little tile. Okay. Good morning once again, and welcome back to this lecture on the fundamental principles, Catholic principles in bioethics. And today's lecture is September 30th of 2017. Let us begin, as always, with a little prayer. In the name of the Father, the Son, and the Holy Spirit. Blessed Trinity, we love you, we adore you, and we come before your presence, becoming more aware that you are with us, that you are always with us. And we ask that you inspire us during this time, that we have to go deeper into the nature that surrounds us and our own human nature, to see us as your image and to glorify you in everything we say and we do. We continue to pray for those who are in great need at this time, Lord, especially the people of Puerto Rico, Cuba, the Caribbean, Mexico, also here in the United States, and wherever there have been natural disasters recently. We pray for your mercy. We pray for human endeavor to go into these places and help out in a humanitarian way. We know that you are with us, and we love you, and we thank you for all your many blessings. This we ask in the name of Christ our Lord. Amen. So we left off with looking at the origin of the universe and the origin of Earth, which we placed about 4.5 billion years ago. Let's see if I can raise that poster again. Origin of universe, National Geographic poster, maybe. Oh, it's not the one I was looking for. Maybe it shows it here, this one. Okay, so that's one poster I'm going to use. And we were looking at possibly 13.5 billion years ago, 13.7, 0.8 billion years ago, the Big Bang, the moment of singularity from which everything expands. First there is an inflation, then there is an expansion. The inflation occurs in billions of seconds, where the first material things are formed, which are the subatomic particles known as what? Quarks, right? Quarks make subatomic particles, the three classical subatomic particles: protons, neutrons, and electrons, particularly protons and neutrons. And then these subatomic particles coalescing in a stable form atoms, and then from atoms all matter that we know today. Then comes that huge expansion in the billions of years. So there are two different time scales going on here, right? And as we say time, we also say space, because of the space-time continuum, which integrates the fourth dimension to the other three dimensions of volume. So to the moment of accretion. And what is accretion? When gravity wins, if you will, which is when matter begins to condense and collapse into what we know as galaxies, and within galaxies, solar systems, millions of them, millions of galaxies, millions of solar systems within galaxies with each one. And typically solar systems will have what around them? They'll be trapping mass, either mass that comes out of them or mass that is flying by, which we call planets, all right? So to witness our own solar system, and particularly planet Earth, which we put much more recently than 13 billion years ago, we put it at What date, more or less? The origin of the Earth? 4.6, 4.5 billion years ago, all right, as a huge ball of gas that continues to condense. And eventually we get a thick liquid called magma with a core of iron that is solid, and then the crust that continues to cool down enough to solidify, and all that process is called accretion. And the crust solidifies enough to have what on the atmosphere and on Earth, on the surface of the Earth, the key substance that is needed for life, for organic life to occur. Right? Think of life as metabolism, and metabolism is essentially a soup that is alive, where chemical processes are occurring by building up molecules and breaking down molecules. And so that soup is going to occur in what medium? A liquid medium of a very interesting molecule called water, right? In the liquid form. So in other words, the Earth had to cool down enough to have water in its liquid form, which we can say in temperature is in the centigrade degree, the centigrade scale is what temperature? From 0 to 100, exactly, which is a very narrow range when we look at the temperatures of the universe. Extremely hot where there is light from the sun, extremely cold where there is no light from the sun, all right? So on the sun side, typically water will be in a vapor form. On the shade side, typically water will be frozen, solid, impossible for the soup to be alive. All right, so you see the combinations of physical and chemical factors to have the possibility of life on Earth. Okay, so we're going to continue to explore and delve into this matter and energy. So the things that are in the universe come in two forms to us that can be measured, and that's what makes empirical science something that can be measured, right? Those two forms in the picture are what? Anything in the universe that we can measure comes to us in one of two forms, and there's an interchangeability, by the way, between those two, the E equal mc squared. So what are those two forms or states exactly? Energy or matter, right? Matter or energy, or mass and energy. But it turns out that that mass and energy that we can measure either with the simple senses, in the middle world that I'm calling, or with instrumentation in the microscopic or the super microscopic world that I'm calling, right? Lost my train of thought. Okay, yes, that is what we call visible in quotations or detectable, measurable mass and energy, right? But there is, and what percentage of the universe does that mass and energy form that we can measure? It's a smaller percentage of this little pie here, remember? It's, well, we can measure gas, right? Yeah. And we can measure planets and stars somehow, right? So that's about 5% of all the mass and energy that is estimated to be in the universe. The other mass and energy is called dark. This should be in quotation simply because we have no access to it yet. In other words, our instruments, they're beyond the limits of our instrumentation today. Just like galaxies were beyond the limits of instrumentation 1,000 years ago, but no longer, okay? So the expectation is that with more sophisticated technology, we'll be able to get into at least the dark matter and also into the dark energy and start expanding this pie. You realize that this little pie here should expand out this way, depending on how long we'll last here on Earth or anywhere else with the advancement in technology. Hopefully there is a concomitant advancement in philosophy and theology and ethics to follow that technology, because that technology by itself is extremely dangerous. It's very sophisticated. I don't know if you saw it. Little aside on the power and technology. Just a couple of days ago, not a week goes by without a bioethical issue, all right, arising, a new one. I don't know if you saw it. I picked it up on the BBC about 2-3 days ago. The genome editing that is happening in China. So they're taking early human embryos and they were able to correct a genetic defect that is called thalassemia, which is a single base pair mismatch. A single base pair of the 3 billion base pairs that make up the human genome, the 3 billion, that's 3 times 10 to the 9, right? 3 billion. One single mismatch, it's called a point mutation, causes a pretty drastic genetic disease called thalassemia. Well, Chinese scientists were able to go into the human genome of an embryo and correct that mismatch, correct that letter. It's like telling you, here's a book, Meyer maybe has 3 billion words, 3 billion letters that make up that text, right? And those are specific letters in a specific arrangement that make words, that make sentences, that make paragraphs, that make chapters, that make the whole book, right? Let's assume that there are 3 billion letters that make up that book. One of those letters is mismatch. There is an error of a G instead of an A in there. In one, only one A, not all of the A's. Only one A in there is mismatch. They went in and they corrected that A for a G at the genome level, okay? Pretty fantastic. In principle, it would cure thalassemia. What's the downside from the bioethical perspective? Of course, they did not do it in a whole human body because we have how many cells? Trillions of cells. And you're trying to change that genome on every cell, right, that expresses. And so you have to go, what's the ideal? To go to the time when we are a single cell, or maybe 2 cells, at the zygote level or at the morula level. Because if I change that one at the zygote level, how many cells are we? 1 cell, which is also known as a fertilized egg, which by definition is no longer an egg because now it's fertilized, which is diploid, has a full genetic complement, right? It's a fusion of the egg and the sperm, but it's still 1 cell. So if I change the DNA on that 1 cell of that 1 zygote of whatever species and allow that embryo then to progress, I have changed the genetic makeup of that individual of that species for life. And if that individual reproduces, that individual is going to pass on the changed genome. Yeah? So it is changing the human genome. And they got human embryos to do that, all right? How did they get the human embryos? They did human cloning. So China is cloning human embryos and is editing the human genome. Who has a right to do that? The human genome belongs to all of us. It's the patrimony of humanity. It's my genetic makeup. It's yours. It's everybody's genetic makeup. The 7 billion that we are. They're doing it. Why are they doing it? Because they can do it. Poder, querer es poder. They can do it, therefore they do it. Totally pragmatic, utilitarian. They decide. They have the technology, they do it. If the dog does not wag the tail will wag the dog. Right? And so bioethics is subtle, but real and important and critical and vital. Now, the countries that have scrupulous and have at least committees, and so today, to this day, human cloning is forbidden in a number of countries. Those countries will then stay behind, will lag on the technology and on the improvement because that will have real medical effects to cure genetic diseases. Okay, and so now there's economics involved in this because they're getting an advantage, an edge on competition. And there's very severe competition of all this going on. There's pharmaceuticals, there's procedures and protocols, clinical protocols and stuff that is going to starting up to millions and billions of dollars. So there's economic pressure. If there's economic pressure, there's going to be political pressure. And the political pressure is going to be toward utilitarianism. Let's experiment those darn human embryos. It's only one cell anyway, and we're not going to implant those embryos. We're going to kill them after we get the result of the experiment, until we get it right, and then we'll market it. In the meantime, human embryos are being killed. You get it? You see the connection? Subtle but real there. It just happened about 3 days ago in China. It's not going back. It's not going back. Okay, so instrumentation, powerful and dangerous. We need the analysis. It's not about saying, let's stop instrumentation and go back to the jungle and the forest. No. Instrumentation is here to stay because it's an extension of our being, and we're not going back to the rotary phone with a wire hooked to the wall, right? But how to use it wisely, respectful of the human dignity and all that, that's the challenge, and that's the rub of bioethics. So moving forward then, if we have the origin of Earth about 4.5 billion years ago, more or less, the crust now is cool enough to have liquid water, but it turns out that the atmosphere is a reducing atmosphere, right? It's not a benevolent atmosphere to life as we know it today. And I want to raise Meyer 3 here, chapter 3, the outline, which is basically what I'm following today. That primitive atmosphere was made up of these 4 main gases, as you know, ammonia, methane, hydrogen gas, and water vapor. And that combination gives a reducing atmosphere. In other words, there was no substantial oxygen there, bless you, and therefore, if there was no oxygen gas, O2, then the atmosphere was not oxidizing, was not a rusting atmosphere, all right? If it's not an oxidizing atmosphere, then it's the converse chemical formula, if you will, or chemical reaction, which is a reducing atmosphere. In other words, it is about accepting electrons instead of giving electrons. The point is that there are bacteria and archaea that can grow in these conditions, all right, can grow in these conditions. Okay, so let me stop there for a moment and give you some of the evidence for life on Earth beginning about between 3.8 to 4.4 billion years ago. These are, let's see if I can close this. These are known as zircon. What is all this? I want this. Oh, worse. Just one at a time. No split. I'm lost it here. I just want, well, let me go back to this. Just want to show the main slide, that's all. Okay. So these are known as zircon crystals, and they're very old, ancient, okay? They go back to about 4.5 billion years ago. And the way we know that is because within, embedded within these crystals is some uranium. And it turns out that the half-life of uranium is about 4.5 billion years ago. 4.5 billion years. Remember, the half-life is the amount of time that it takes for half of the material to decay, the isotopes to decay to a lower isotope, okay? And for uranium is about 4.5 billion years, which is a very handy number to date the Earth, because then we can say that the Earth, that carbon was around at the surface of the Earth. And remember, carbon is needed as one of the 2 elements for organic molecules, right? And the other one being hydrogen. Now, the atmosphere at that time had plenty of hydrogen gas, so the 2 elements are there necessarily physically for life. Okay, moving forward then, this is in Australia. Some rock formations have been found, and these rock formations are called stromatolites. Stromatolites. Let me see if I can maybe. Father Dayan, if you can hear me, we're getting feedback from a microphone in your computer. I wonder if you can mute your microphone until you need to speak. Because if I, okay, thanks. Because if I mute it from here, then we cannot know when you want to say something. But if you mute it, then you have control. Thanks. Okay, so stromatolites are these boulder kind of formations on the coast of Australia. And when scientists did cross-sections of these stromatophytes, it turns out that they found these layers. And going deeper into the microscopic structure of these layers, these are actually layers of organic material. It is sedimentation. It's estimated that these are bacteria or archaea that form these layers. Remember how I talked about cycling in one of the 7 steps for life? The cycling that can happen, one type of cycling is the daily cycle between light and darkness, but another type of cycle that would affect mostly what today, do you think? Well, many living things, but the ones that are most dramatically affected by the cycle, the 24-hour cycle of day-night. Day-night, think of light, no light. What is mostly, what life? The plants, right? Plants for photosynthesis or not, okay? But then there's another cycle that can occur on a yearly basis, and that's going to be a seasonal cycle, which depends how close or far away the organism is from the equator, because at the equator there's going to be practically no cycling. The day and the, aside from the day and night being 12 hours and 12 hours, the other thing that it's mostly the same temperature year round. But as we go away from the equator, either north or south toward the poles, the temperature is going to change more and more dramatically, right? And somewhere in the mid part between the longitude 30 and 60 degrees is going to be the temperate region, where typically we get how many seasons? 4 seasons: spring, summer, fall, winter. And so that's a cycle. It's a temperature cycle, aside from the light cycle. So the different types of cycles, all right? More dramatic on the poles, it's going to be mostly cold. But the cycling could have produced this layering, all right? The point is that there is organic material here as residue. And so it is estimated to be from bacteria or archaea. And I keep mentioning these 2 groups of organisms. They are, remember, 2 of the 3 domains. What is the 3rd domain? The bigger picture of the classification of living things? Eukarya. Eu meaning true. Karya is a reference to the nucleus in Greek, so they have true nuclei. These are prokaryotes, meaning that they don't have an organized nucleus. They don't have an organized nucleus. Typically, there's an area of concentration of DNA, all right? There is an area of concentration of DNA, but there's no true nucleus, meaning that there's no envelope of the nucleus, there's no membrane of the nucleus, all right? So this is the only membrane that is around for these creatures is the cell membrane itself, to maintain the soup occurring, to maintain metabolism occurring within this one compartment. The membrane, remember the phospholipid membrane, phospholipid bilayer, which on the outer side is hydrophilic, and on the inside is hydrophobic, right? So it's a self-organizing membrane because of its own polarity and how it reacts to liquid water. At any rate, I wanted to point out the main difference between these 2, archaea and bacteria, which is at the level of domain, which is the largest possible separation between living organisms, okay? It's a pretty radical difference. Comes down to a microscopic layer within the membrane that is called peptidoglycans. Peptidoglycans are mostly carbohydrates, one of the four main groups of organic macromolecules: carbohydrates, lipids, proteins, and nucleic acids, right? Well, peptidoglycans are pretty complex carbohydrates, and they are sandwiched in between the outer and the inner membrane of the cell wall of bacteria, but not in archaea. In other words, this layer of peptidoglycan, which is present in all bacteria today, is not present in archaea. And that's why they're called archaea, because it's a reference to being archaic, ancient, old, right? Someone calls you archaic, you know what they're talking about. So the fact that they lack that membrane there, all right, or that layer, makes them older than bacteria. In other words, that is assumed that that layer developed after the archaea. At any rate, these two together are prokaryotes, and they're definitely microscopic, and they're also very tenacious, meaning that they can live in extreme environments. We're trying to get at a reducing atmosphere here that can actually thrive, not just survive, but thrive in a reducing atmosphere. They love methane, they crave ammonia, all right? And those are called extremophiles. Remember, philia is a reference to love, phobia to hate. So they love extremes. For example, halophiles or halophiles, depending if you're American or Brit, is a reference to salt. So these live in extremely salty pools, extremely salty environments. And here's a macro photograph, electron microscopic photograph of a bacterium that lives in extremely salty environments. Like, for example, the Dead Sea. I don't know if you've ever been to the Holy Land, and the Dead Sea is called dead. It's because it's about 10 times more salty than the ocean. You've been to Miami Beach, and you take a little bit of salt. If you open your eyes on the water, it's going to stink for a little bit. It's salty, right? All right. Now, make that 10 times saltier. All right? 10 times. You increase the amount of salt 10 times. It is so salty that if you open your eyes in the water of the Dead Sea, it will just burn your eyes out. If you have a little cut on your skin, you will feel that cut a lot. It is dense. It is so salty that it's actually thick. You know baby oil? It's like baby oil, the thickness. It's not oil, it's water, but it's very thick. So people can float standing up. All right, it's that thick. It's just buoyant. And so nothing lives there except bacteria. So it's very clear water, and nothing lives except bacteria, extremophiles, halophiles. Andrea? Okay, but others like extremely hot temperature, like living next to geysers where the water is almost boiling, okay? And there are thermophiles, they love heat. There's some micro photographs of them. Others are hot and dry, like in desertic environments. Those are known as xerophiles, zero as a reference to dryness or desert. And then there are others that actually are resistant to radioactivity, like UV, ultraviolet radiation. And they're called just radio-resistant. And they can be zapped with radioactivity, like the microwave, and they'll survive and thrive. So these are extremophiles. Some are archaea, some are bacteria, but just to say that even today we have examples of living organisms that can tolerate a reducing atmosphere. One of them is called a cyanobacteria. Now, cyanobacteria, cyano is a reference to a color, cyan. What is the color cyan? Blue-green, right? Okay. When I was studying, I think I mentioned this, biology at FIU in the 70s, last century, these were still classified as algae. They were known as blue-green algae. Blue-green algae. Today, it is known that they are not algae at all. They are bacteria because they have mostly the membrane of bacteria, but they do have chloroplasts and they photosynthesize. Or at least not chloroplast as such. I take it back because the chloroplast involves a phospholipid bilayer membrane, which these guys don't have, okay? But they do have the thylakoids, and the thylakoids, excuse me, are the little corpuscles that contain chlorophyll pigmentation. So the bottom line about the cyanobacteria is they can photosynthesize, and that's why they are this blue-green color, all right? Because those pigments absorb on the red side of the spectrum, and therefore they reflect the blue light, which is what is seen, or the green light. That's why we see plants as green, because they absorb on the other side of the spectrum, on the light spectrum. Here's a microphotograph of them, and you can see clearly the pigmentation, the green pigmentation. And they have been dated back to about 3.8 billion years ago. And they went on for about 1 billion years, and very successfully. And what is the big deal about these little guys? It is estimated that they are the ones who essentially produced the bulk of the oxygen on Earth. So transforming the atmosphere from a reducing to an oxidizing atmosphere, which now turns an atmosphere that used to be toxic, extremely toxic for every other living creature, into a benevolent atmosphere. Something that we can breathe, because we're going to be respiring, and in respiration at the cellular level, it's an oxidation reaction, and we need oxygen, okay? Moving forward, here's an example. How do we go from prokaryote to eukaryote? Basically, it's a membrane issue. Remember, membrane within membranes, that's one of the steps on the life passage, the origin of life, to generate membranes within membranes. One of the theories that is around, the prevailing theory, is that there was some kind of a symbiosis between maybe archaea and bacteria, and maybe the cyanobacteria were one of those bacteria that was absorbed by an archaea or by another bacteria, but managed to survive within the other bacterium. And that gives the new bacterium the possibility of photosynthesizing through the cyanobacteria that was absorbed, all right? And eventually that cyanobacterium, being inside a host cell that is supplying all the nutrients, may be degenerated just down to what is known today as the chloroplast, but maintain the phospholipid bilayer. Maintain the phospholipid bilayer. A similar argument could be made for the nucleus. The point is that the DNA was already there, or maybe even further back, RNA, but we observed that today, all of the eukaryotes have, by definition, a membrane nucleus, an enclosed nucleus, so somehow that membrane had to be generated. Here's a bacterium that has an elaborate membrane system. This is actually an extension, if you can follow here. Here's the cell wall. Actually, bacteria have an outer membrane and an inner membrane. Okay, here's the bacterium cell wall, but if you follow the inner membrane, you see how it curves here and protrudes out into the finger-like projections. And the same back here is follow the inner membrane, see how it bends in and forms this envelope here. Okay, so this could be the possibility of a beginning of an inner membrane. This would be a single layer, of course, whereas the nucleus has a bilayer, and therefore it's thought that at least the nucleus, the chloroplast, and the mitochondria came from an endosymbiosis. In other words, absorbing other bacteria or archaea that survived the absorption process. So this is a current bacterium. The name is Gymnata oscuriglobus. Okay. And there's a scale of 500 nanometers, just to be clear. 1 nanometer is a millionth of a millimeter, and a millimeter is going to be about the thickness of a nail, maybe the thumbnail. Okay, that's about a millimeter thickness. Well, 1 millionth of that, right, 10 to the minus 9 is a nanometer. So that's the scale there. You can see that these are definitely microscopic. That's an electron micro photograph. But anyway, we have examples even today of membranes within membranes occurring in prokaryotes. So the current theory is by endosymbiosis, we go from prokaryotes to eukaryotes. Again, these are current diagrams of eukaryotic cells. And the two fundamental eukaryotic cells are either animal cells or plant cells. The main difference between the two, as you are going to be what? One has, so both have a nucleus, right? Both have at least one nucleus. Some cells are polynucleated, like muscle cells, but all the eukaryote cells have an organized nucleus with a phospholipid bilayer, just like the cell membrane is a bilayer, right? And both, so what's inside the nucleus again? The DNA or RNA, but typically the DNA, which is the genetic material, is the information, is a storage, and that's well secured, okay? It's well secured, that information that gets passed on from generation to generation. Both types of cells then, plant cells and animal cells, have a nucleus, and well-organized nucleus, very similar to each other. And both have also the other organelle that is essential for life, the mitochondrion, right? What do mitochondria do? Yes, they are involving recharging ATPs at the chemical level. ATP is the chemical molecule of the cell. Adenosine triphosphate is like the little batteries that are needed for the flashlight, right? So that battery, if you turn on the flashlight, the battery is going to run out of energy. The battery needs to be recharged periodically, and it's recharged through these organelles called mitochondria. This is where the oxidation reaction occurs. It's an oxidation reaction, part of what is known as the Krebs cycle, etc. It's metabolism. It's metabolism at work, and it occurs within this mitochondria, which again is a bilayer. There's an outer layer of the mitochondrion, which is smooth, and then there's an inner layer that is corrugated to maximize the surface on the inside, and that's where the actual reaction occurs. My point is that since these mitochondria are involved in supplying energy to the cell for metabolism, then they occur in both plants and animals. Okay, the misconception is that because plant cells have chloroplasts, then they don't have mitochondria. No, both plant and animal cells have mitochondria because both are alive, both need energy for metabolism. Follow? But then the extra thing in plants is precisely these chloroplasts, which could be derivatives of cyanobacteria that survive an endosymbiotic event. See? All right, so that gives the advantage, if you will, of plants to be able to photosynthesize, and that makes them producers in the world, in the living world. And producers of what specifically? Producers of food, a type of food that the animals are going to consume. So the animals are consumers. We don't photosynthesize, right? We don't produce our own food. And at the chemical level, what food are we talking about? What is being synthesized in the photosynthesis? What is a carbohydrate, and it's called glucose specifically, okay? So glucose is what's being synthesized. That is the essential food, which is also for the plant because the plant is also alive, and therefore the plant also consumes glucose, but it produces an excess of glucose, which can then be consumed by the animals. Okay, so at least 3 main organelles are needed for the eukaryotes, for the eukaryote cell, which is the nucleus, the mitochondria, and the chloroplast. And you understand that this is a quantum level of sophistication, a prokaryote from a eukaryote, right? So a prokaryote cell, it's much more organized and developed, and can live longer, and also is amenable then to combining with other similar cells to form tissues. And then those tissues are also amenable to combining tissues that have a similar function to form organs. And then organs that have similar functions to combine also into systems, what we call organ systems, of which we have about a dozen, depending on how they're classified. And then organ systems of different function, put together within one bag, we call the organism. And the one that coordinates all that, there's typically at the higher level of organisms, there will be one of those dozen organ systems that coordinates the others, that tells the others what to do, when to do, and how to do it. That is going to be the nervous system. And the others, what are examples of organ systems in our body? Digestive, reproductive, urinary, circulatory, respiratory, right? All those, see, they have different function. If I lay out an intact respiratory system here of a human being, and then on this other table, an intact circulatory system, and over there, an intact digestive system, but they're laid out differently, is that a living body? No, they have to be put together and they have to coordinate, right? So each one of those systems does the right thing at the right time. And it's actually a combination between the nervous system and also what keeps the whole bag together physically, physiologically. What's the outside of the balloon? The skin, which is another system. It's a whole system that is called the integumentary system. The integument is the skin, right? It's interesting that both the nervous system, which is at the innermost part of the body, and the integumentary system, the skin, both come from the same germ layer in the early embryonic development of the three germ layers, endoderm, mesoderm, and ectoderm. It is intuitive that the skin will come from which one of those three layers. If they're named according to their physical position in the early embryo, endoderm, mesoderm, and ectoderm. The intuition is that the skin comes from the ectoderm, right, on the outside. What is not intuitive is that the nervous system, which in the developed organism is in the center, in the innermost part of the body, also comes from the ectoderm. So the skin and the nervous system have a common origin in embryonic development. Interesting, because there's a pouring in of the ectoderm down the central groove of the early embryo. It's called an invagination process. It's a pouring in of these cells that form the neural tube and eventually differentiates at the head region into the brain and the rest as the spinal cord. And then the branching nerves out. So all this in embryonic development done within 9 months or less. Exciting. Forward. All right, so examples, then there is one grab bag that is not used too much today anymore because it's too simplistic, but they call the protease. When we talked about the kingdoms, when we went down from domain to kingdom, right? One of the kingdoms is this protease, and I say it's a grab bag because it includes unicellular organisms and also colonial organisms and also multicellular, truly multicellular organisms. Some are producers like the brown algae here and the red algae. In other words, they photosynthesize. And the euglena, for example, at the unicellular level, and these colonial, these are called volvox. And some are consumers, like the amoeba, the paramecium, etc. And these other colonial protists. But my point here is that there is this colonial variety, all right, which are not unicellular, but they're not truly, there's multicellular, but in a loose way, these cells, these organisms are called colonial because it's a loose association of cells, but the there is a distinct body that defines the organism for sure, but typically it's just 1 or 2 layers thick, 1 or 2 cell layers thick, and the interaction of the cells is just by close proximity, mostly interacting just with the cells next to each other, adjacent cells, neighboring cells, but there are no true tissues yet in these colonial organisms, and that's why they're not truly fully multicellular as others are, but they're still protists. They're small, typically they're microscopic, and this early ancestors of these protists could have been a transition from unicellularity to multicellularity through this process of known as colonial organisms. I want to give you another example that is living today. Ever been to the beach and gotten stung by a Portuguese manowar? Nasty. Okay, these are coelenterate, meaning that they belong to this group, or cnidarians, just like jellyfish and sea wasps and other creatures, anemones, and the coral. They're hard corals. The typical coral that we know is a hard coral, which has a calcium carbonate shell, right? But there are also soft corals that have mostly a protein skeleton, and that's why they sway back and forth in the ocean. Soft corals, they're also known as sea fans and sea whips, etc. Anyway, corals are also coelenterates. They belong to the same phylum of animals. These are animals, beautiful to look at, but deadly. The tentacles have little cells called nematocysts, and inside the little nematocyst is a toxin, and the toxin is a neuroparalyzer. Those stinging cells, the neuroparalysis, what they do typically, they extend, and these tentacles can extend 10 to 20 feet away from the bubble. So you see the bubble floating over here, and the tentacles, depending on where the current is going, the tentacles can be 20 feet away from that bubble. All right, so if you see the bubble, stay clear away from the bubble. What they do is these nematocysts have a little trigger, they fire away, they sting, they actually inject a little needle into the victim and paralyze the victim alive, typically little fish, okay? They paralyze the fish alive, and then the tentacles move up into this region, this mass, and this mass exudes digestive enzymes, digestive proteins, which digest the fish, and then other masses of cells here suck up the nutrients that are being broken down by the digestion. It's a type of external digestion, and then there's some kind of loose, open circulatory system inside where the nutrients are then squished around through the tissue, eventually to feed the entire organism. But what I want to point out is that this is not truly a single organism, as beautiful and as integrated as it looks. It's actually a colony of tissues. It's a colony of specialized tissues that together form the organism. And it is defined as a single species, okay? It is defined as a single species, and this is one individual. However, it's a colony of tissues that have come together in a specialized fashion to together live, be able to absorb nutrients, and also be able to reproduce. So some of tissues in here are specialized cells that become gametes. And then there is, of course, an external fertilization whereby they may even have isogametes, meaning that they are, it's no differentiated into male and female. They have the same sex, if you will, but when these gametes come together, they form a diploid cell. And then typically all of these organisms that live in the ocean, have many stages of development, and the larval stages are typically microscopic and unicellular for a while, then they become multicellular but still microscopic, and they form part of what is known as plankton, and eventually they will continue to grow and develop into the adult. But they go through many stages. Even crustaceans and arthropods like crabs, lobsters, shrimp, and all those pre-peak crawling creatures. The lobster, I think, goes through about 10 or 11 larval stages, microscopic. Before we see the actual lobster, that's the adult. Mm-hmm. And the larval stages, of course, look nothing like the adults, not at all. That's why simple morphology is not enough to classify them. We have to go into the DNA and see what is the phylogenetics, where do they belong on the tree, and what is the homology, so that we can start making relationships, branching of the tree. Okay? You follow me? The language is okay? Okay, we are coming up to the three main kingdoms of eukaryotes. So we've gone from protobions, which is the beginning of a metabolic process, into forming what we call today archaea and bacteria. The prokaryotes, definitely unicellular, but also simple in their internal organization. And then from prokaryotes to eukaryotes, meaning membranes within membranes, and the three big, so that we can have organelles, in other words, specialized compartments within the cell that do different functions. And the three main organelles that were needed initially to go into eukaryotes was, again, the nucleus, mitochondria, and chloroplast, right? And then, whether the chloroplast is present or not, we can distinguish between plants and animals. And we're getting more and more complexity of life with long periods of time. We're into the billions of years here because we started around 4 billion years ago, right? Now, today, on the eukaryote side, then we have the three main kingdoms: plants, fungi, and animals. Plants, beginning with the most simple ones, bryophytes. These are otherwise known as mosses. The mosses, okay, they don't have a vascular system, and that's why they have to stay small. To the other end of the complexity, so this would be the simplest plants today, approximately, to the most complex plants are the dicots or eudicots, the flowering plants, and the flower is actually specialized leaves. So imagine, including the stem, the anther, and the stamen, all the different parts of a leaf, of a flower, as beautiful and varied as they are, those are specialized leaves. Imagine the evolution that has occurred there. Mm-hmm. To the point that some parts of the flowers do photosynthesize. Why do you think that orchids, flowers, stay for 2 or 3 months? And they have color, therefore they have pigments, therefore they photosynthesize.
Isn't that the Ponciana?
Yes, this is, right, correct. This is the flamboyant in Spanish or French. It's a Ponciana, yes, they're beautiful. They got hit by the hurricane. They have very tiny leaves. They're very messy, because they drop all this orange that you see here, they drop it down here, okay? But they're beautiful, huh? They look like when they flower, they're like in flames, they're on fire, and that's why that's the name in French, flamboyant, it's on fire.
It grows below,
Sorry?
Grass cannot exist under that.
That's right, and I think it has to do with all the droppings of the rotting flowers, and maybe there's some chemical composition going on there that prevents.
You say are leaves.
They're adapted leaves. They used to be leaves. They're adapted, but the origin of flowers, of all the parts, the petal, the sepal, all the different parts of the flower, they are adapted. In fact, the sepal, which is the lower part of the flower, is still green, and then on top of that is the petals.
The flower also has a little pod in it.
Yes, that pod.
Liquid in it.
Yes, the pod. Yes, the liquid is the nectar. The pod is the pistil, and inside the pistil that's the female reproductive structure. Inside are the ovary. Is the ovary at the base is the ovary, and inside the ovary are the actual eggs, the ova. And then the male part is the stamen that comes up like this, and the part that stains yellow. That yellow is pollen, and that pollen is the sperm of plants. In fact, 2 sperm cells inside each pollen grain. So that's the equivalent, that's the plant equivalent, and so plants also reproduce sexually. Of course, they don't have copulation, but they do have fertilization. Okay, So these reproduce. So the flower is the evidence that plants also reproduce sexually. In other words, there's production of sperm and there's production of egg, and the fusion of two to make zygotes. So they also have zygotes.
There are also some plants that are just male and just female. Is that true?
Yes, yes. In fact, in pines it happens. There are most of the pine cones that we see. The large one is a female cone because inside is a little egg. All right. The male cone is a smaller one. It's longer and it's more fuzzy and it's not as visible. But there are male cones and female cones. Mm-hmm. And some pine trees have both types of cones, and other pine trees only are differentiated so that there's a male pine tree that only produces male cones, and then the female pine tree that produces female cones. Our slash pine here has both, produces both the male and the female cones. But there's still cross-fertilization into other individual trees of the same species. So there's still cross-fertilization.
So why is it that some fruit trees don't bear?
Sorry?
Why is it that some fruit trees do not bear?
Don't bear fruit?
Don't give fruits.
Yeah, well, it has to be with a combination of factors, typically climatological factors. But some plants, even though they are flowering plants, there's another mode of reproduction for plants that is very effective, that is called vegetative propagation. Like grass, for example, it puts out little shoots, and from the shoot, a new tuft will grow. For example, the lawn grass, if you pull out a tuft of grass on the lawn, you'll see all these little shoots that go in and along the way. That's vegetative propagation. That's asexual reproduction. It's essentially a type of cloning of itself. Or sometimes it can happen by little buds underneath, like pineapple or orchids and bromeliads, right? They put out little buds on the side. That's a different type of reproduction. It's called asexual. It's one of the two modes, asexual reproduction. It involves less energy. Generally, overall, it only costs the plant half the energy to reproduce asexually than to reproduce sexually. So it has an energetic advantage, but the disadvantage is that there's no genetic mix. It's a clone of itself. So as long as the environment stays the same, they're doing great. But if the environment changes, especially a little more drastically, there's not as much variation. So that's the downside of asexual reproduction. It doesn't have as much variation, which is actually going to be the topic of next lecture. It's a good segue. We're going to look at variation in nature, all right? And so start looking at, you should read, don't summarize yet, but read chapters 4 and 5 of Meyer, okay? And finally, the animals from, like I mentioned, Cyanobacteria. Here is a hydra, which is a type of anemone. This is the adult phase. The embryonic phase, the larval phase was free swimming and didn't look anything like this. But these are Cyanobacteria or cnidarians on the simple side of the animal kingdoms, all right? To the most sophisticated ones, mammals and marine mammals, which are even more sophisticated, more evolved, I should say, in this sense, that marine mammals. So you see, this is it's a blue whale or humpback. I think it's a blue whale. It's the largest. It's a blue. Yeah, it's the largest mammal on Earth, and you see, it has tail and flippers and so forth. But if we were to take an X-ray of this humongous flipper, that would probably be the size of this room. We take an X-ray of that. What do we see? We see digits. We see bony, the bony structure of fingers. All right, so this is the remnant of a foot. And then if we took a photograph on the backside region here, we would see the remnants of a pelvis, which is pelvis is used for connecting the lower extremities, the legs. So in other words, this creature, the ancestor of this creature used to be a tetrapod. If it's a tetrapod, it means that it walked. Where? Not on the ocean, but on land. But the tetrapods, turns out, came from the ocean originally, from water. And so what I'm trying to point out here is that marine mammals derive from terrestrial mammals, and the terrestrial mammals themselves derive from reptiles in the ocean. So talk about evolution and development, all the way from cnidarians or coelenterate, like even sponges are more simple than cnidarians, and they're animals. Sponges are not plants; they don't photosynthesize. Okay, but they are animals. All the way to marine mammals, which have evolved back into the ocean. But still, on this, we take another X-ray here in the chest area. What do we find inside? Any gills on this creature? Any gills? No gills, therefore no fish, right? But it has to breathe somehow. So what does it have inside? Lungs, big, humongous lungs. And if it has lungs, it has to breathe. And is it going to breathe inside the water? Too little free oxygen to breathe inside water, right? It's mixed in the water, so it has to come to the surface to breathe. And so it's convenient for the nose to have migrated to the top of the head, so that for breathing, it only has to bring out the nostril or the blowhole, and that's why when the whale comes up to the surface, it goes, it makes a little spout of water. It's clearing its nose so that it can breathe air, take a gulp of air, big gulp of air, fill its lungs, and then go down for half an hour, 1 hour, or 2 hours, depending, and then come back up again. So talk about adaptation. The blowhole is the nose that has migrated all the way up to here to live in a marine environment, but it's a mammal. What else? Okay, so here's just a diagram, a cladogram, or a phylogenetic tree, right? Showing branching, beginning with the prokaryotes way down here in the Precambrian era. We're going to look at the geologic eras in a minute on the back side of this handout of chapter 3. Before I get there, let me make sure I did the passage from prokaryotes to eukaryotes, from eukaryotes to multicellularity. Sorry, over here. And then one evidence of eukaryotic metabolism is the production of lipids, and that is estimated to have occurred about 2.7 billion years ago. And there's some fossil evidence of lipids that date this back almost 3 billion years ago. So the complexity of life happened fairly early on. There was a boom, an explosion of diversity of life. And these processes are called, in common language, explosion, but in scientific terminology, it's called radiation. Radiation from one common ancestor, several descendants develop, several different species develop. And those processes are called radiation. And typically, they tend to match drastic geological events, like volcanic activity in a whole region, or the ice ages, the freezing and the thawing of water on certain parts of the earth. Those dramatic climatological events typically have an extinction process, and then after the extinction, a radiation process. So their cycle, they follow one another. And why? Because at the extinction level, what happens is many species that are voracious are eliminated. And so now there's room, literally there's room for other new species to develop, whereas before they didn't have a chance because the previous species were so voracious that they would clean everything in sight. And that was the classical case of that was the dinosaurs. I'll get there in a minute. I just want to make sure that I've covered the different steps here, the 3 branches. Oh yeah, I didn't mention too much about fungi. So let me go back to fungi for a moment, because people are not that familiar with fungi. Fungi is the 3rd type of kingdom, and they tend to look like plants, but they're kind of funny looking plants, right? Why do they look like plants? What makes them similar to plants? The most obvious thing is that they look like they are rooted on the earth, right, on the ground. They're not really rooted, even though they do throw down some system of of cells called hyphae and projections that go into the ground to absorb nutrients, rhizoids, but they don't have chloroplasts. If they don't have chloroplasts, they don't photosynthesize. If they don't photosynthesize, they're not producers, but they're living. So they have to be consumers. But they're a particular type of consumer. It's a consumer that feeds on decay organic matter. That decay organic matter could be plant or animal, right? Anything that dies, or could be other fungi also that died. So that makes them saprophytes. I talked about saprophytes earlier, okay? That means that they feed on decayed organic matter, and that's a good thing, because otherwise, like together with bacteria, they break down dead plants and animals and fungi and bacteria. Otherwise, we would be up to the ceiling and beyond with dead organic material here on Earth. Because plants and animals and bacteria and fungi, it's all dying all the time, right? Someone has to eat that and digest it. Okay, so fungi do that great function. They can be from microscopic, like yeast, which can also cause infections because they're trying to feed also, they don't only feed on dead things, they can also feed on living things. Some of the fungi, like if they try to feed on your toes, then that's athlete's foot, okay, which is a fungus. Or they can be as huge as this is an exception, but that's definitely a mushroom there, okay? And typically mushrooms are much smaller than that, but they are definitely visible. And the arrows here don't mean that from Hydra we get whales, or from yeast we get gigantic mushrooms, or from moss we get flowering trees. No, the dash means that there's a variety, there's a range, but that certainly mosses are much more simple, organically speaking, than flowering trees. And that yeast, to begin with, they're unicellular, are much more simple than mushrooms that we can see with plain sight, which are definitely multicellular. And that hydras are much more simple in their structure than marine mammals. All right, so the arrows doesn't mean a derivation, but that the range of from the most simple to the most sophisticated plants, animals, and fungi that we find today, that all adds up to variety. Variation, and variation between species, but also variation within the species. Different sizes, different colors, different functions, or different capacities for surviving. And it's that variation that evolution, that natural selection is going to act on, is on that variation. So, bottom line, we can develop a phylogenetic tree by looking at similarities of bacteria and archaea, similarities of eukaryotes, the different types of development that they have, and how they are similar to each other to create these branching phylogenetic trees. And we can go back on the tree, hypothetically, to a common origin, which would have been in the early Precambrian, some kind of unicellular organism. This is a more elaborate phylogenetic tree based on molecular evidence. Dozens of, there are about a dozen major phyla here. Today we have about 3 dozen phyla, including the major ones and the minor ones. Minor meaning that they have few representatives, few species in them. Others have a large amount of species. Here are chordates. Anyway, you can see that this is a more elaborate tree, but again, if you go back in time, you find nodes or points of common origin, okay? These are known as common ancestors, these nodes on the tree. And if we go back on the tree, we continue to find commonality at a smaller percentage. So we would consider that 2 different orders of arthropods are more closely related, have a higher percentage homology than this species of arthropod with this species of sponge over here. They have their nodule connection is simply at the level of metazoan, meaning that they have more than one cell or that they're macroscopic. Okay? So the nodules, where they are denotes on the age scale, tells us the percentage homology or the percentage of relatedness in the different species. Okay, so just to show that phylogenetics is today done down to the molecular level and is very detailed. So when we look at phylogenetics, we find a physical, material explanation for it, of relatedness and descent from common ancestors, which then does not need the theory of what was known as spontaneous generation. This proves the theory of spontaneous generation as too simplistic, all right? The theory of spontaneous generation is that, for example, was based on not just simple observation, but what I call naive information or naive observation. A naive observation was this: I take a piece of meat and leave it on top of the counter and let it rot. And eventually that meat turns into flies. You know, the meat turned into flies. That was a classical example. Look, this meat, I just left it here, and now the meat turned into flies spontaneously. Overnight, the meat became flies. Dead meat from another animal became fly, which is a type of insect. Okay, that was known as spontaneous generation, and it was kind of a magical way of looking at the origin of life. Today is not only simplistic, but actually naive, because then someone had the bright idea of taking that meat and putting a mesh wire around it so that the flies could not get into it at night and deposit the larva, and the larva would not then grow into the adult fly, okay? And when the piece of meat was left with a mesh around it, lo and behold, there were no flies generated, okay? So somehow the fly that was landing on that meat was doing something to the meat to generate new flies. Yeah, what it was doing, it was depositing microscopic eggs that could not be seen, and the larva was eating the flesh inside that eventually that larva turned into adult, and there the flies started emerging, the adult flies started emerging from the rotten meat because the larva was eating inside. So the naive observation didn't work when we get into the scientific method of observation and experimentation and so forth to the level of sophistication that we have. Don't have time now, but you can look at this video on Wikipedia. I send you the entire presentation. So you look at it, it explains spontaneous generation. And the one who disproved that was Louis Pasteur. One of his great achievements, by the way, practicing Catholic. He used to pray the rosary every day. And Louis Pasteur was one time, I can't resist. I'll tell you a little anecdote about Louis Pasteur. He was, you know, Louis Pasteur, right? He invented pasteurization and saved a lot of people from getting contaminated with, at first it was the wine industry in France that contracted him because the wine was going sour, precisely by contamination of the bacteria. You know, it's a perfect soup for bacteria to grow in because you get glucose, you get sugar, turning into alcohol. So what Pasteur did was he invented this pasteurization, which was bringing the wine up to, you couldn't boil the wine. People knew that if you boil water, you get rid of the germs or the microorganisms, but if you boil wine, you ruin the wine. So the pasteurization process basically is bringing the temperature up, but just under boiling point, just under boiling, and it works for milk also. But you leave it there for a longer period of time is what you do. To bring it just below boiling point so that the liquid doesn't boil, whether it's wine, water, or beer, or milk, all right? And you leave it there for a longer period of time. And it still sterilizes the liquid. That was one of his great inventions. Another one was to disprove spontaneous generation, and the experiments are described in that link, so go and take a look at it. He did it in 1859. What's 1859? Publication of The Origin of Species by Darwin. So it's a lot of science that was going on in Europe at that time, you know, rigorous science. But a little anecdote about that people didn't know about Pasteur. It's narrated that he was once traveling into Paris on a train, The trains were very famous back then. It was the beginning of the Industrial Revolution and all that. And the military was also very powerful. And there were soldiers all over the place. They were maintaining peace and all that. And so there was this old man in one of the wagons of the train, right? One of the cabins. There were seats, I think it's 3 or 4 seats over here, and then 3 or 4 seats in front. They sit like that, and for hours riding on the train. And this old man was there praying the rosary, and then in front of him was a young soldier, and the young soldier just kept deriding the old man for being antiquated and praying the rosary and superstitious and all this. And today, the soldier was going on, today is the age of the military, and it's the age of scientists, and it's the age of progress, and it's the age of the Industrial Revolution, and the Prometheus man, man by his own power, look at everything we have achieved. And the old man didn't say anything, was just praying the rosary. When they got to the station in Paris, the old man just shook the soldier's hand and handed him his card, the card of the old man. And the card of the old man said, Louis Pasteur, president of the Royal Academy of Science in France. Okay? So, like Mendel, right? A monk who discovered genetics. So, the semi-modification is possible if we combine Charles Darwin and Gregor Mendel and all the others who have followed into what we know today, the scientific method and the process of evolution. Oh yes, then you can just look at this chart in more detail a little later on, but here I give you just a simple view in perspective of geologic time. If I make it too big, I lose the scale. But it's starting with the Precambrian. This goes into the billions of years, but for the 4 billion years, when the big line here is from the Precambrian to the Cambrian. And there was a mass extinction there that happened about 500 million years ago. 500 million years is 500 million years. Focusing in on this first boundary here. Mass extinction that allowed then for an explosion known as the Cambrian explosion, and scientifically known as radiation. And that radiation was the generation of a large number of new species, mostly in the ocean. And from there is also approximately the transition from prokaryotes to eukaryotes, or regularly the origin of eukaryotes, because the prokaryotes never disappeared. All right, origin of eukaryotes, and then into plants, and eventually the animals, insects, and then we get into the chordates or vertebrates, first fish, then amphibians, which can live, the larval stage is aquatic, the adult stage is on land, eventually reptiles, especially with the development of scales so that they don't dry out, and then the radiation from reptiles, significantly birds on one side and mammals on the other side. So we can see feathers as very sophisticated scales, and also hair as a derivative of scales. The same origin, organic protein origin of keratin. So we get into, that was in the Paleozoic, and we have another mass extinction about 250 million years ago, all right, 250 million years ago, mass extinction, and therefore another radiation into the Mesozoic. It's the age of the mammals and birds, but small mammals, very small, because the reptiles went haywire and went into what kind of, it's called runaway evolution, all the way up to the dinosaurs. In other words, the reptiles were so successful on land that they continued to grow and become more and more aggressive. And they were all carnivore. Well, no, take it back. Some dinosaurs were herbivores. But many of them were carnivores. But the common factor that they had, the common element, is that they became huge in size. And that's called runaway evolution, because there was basically no one, they had no other competition. All right? And so there was another mass extinction here, which happened about 65 million years ago, 65 to 70 million. It's been adjusted to about 65, which is attributed mostly to the meteorite that landed in the Yucatan Peninsula that is buried down there several miles into the earth and caused a humongous explosion that caused such a thick cloud of dust on the atmosphere that the sun was blocked out for several centuries and started killing a lot of the vegetation, which killed the primary consumers, which then exhausted the food source for the dinosaurs, and they started dying because they had such huge bodies that they needed feeding all the time, and there was not enough food. Essentially, they starved to death, which gave, at the same time, that cloud also caused a change in the weather dramatically to a colder environment, and so the better adaptation was mammals, where we become endotherms. We can hold our own temperature, and the hair has a lot to do with that because the hair maintains a very thin layer of air around us, a veneer of air that maintains our body heat. And today we have lost a lot of hair because of the clothing that we wear, but essentially the animals, the mammals, that's what the hair does. The hair maintains a layer of air around the body that maintains that body warmer than the rest of the atmosphere, okay, the rest of the environment around them. So this last mass extinction allowed for a radiation in the Cenozoic, the early geologic era, the past 60 to 65 million years ago, to the mammals and the large mammals. And then we get huge mammals, like the mammoth and the giant sloth and the saber-toothed tiger, which could have been the size of this room, and those giant deer that also lived up in the northern regions that have racks that are immense. All those are extinct today. We see their fossils. But also some of those mammals went back into the ocean because they got so big. Those would be the marine mammals. Eventually we get the primates, and about 2 million years ago we get genus Homo, of which we know at least about 6 to 8 different species of Homo from the fossil record. And the only living species of Homo today is Homo sapiens, you and I. All right? That story to be continued. But this is very fast synopsis and overview, starting with the Precambrian and the prokaryotes, some common ancestor way back here about 4 billion years ago, speeded up to the present variety of the estimated 10 to 20 million different species existing on Earth. So in the big picture, radiation upon radiation upon radiation. Okay. I know I've sped through this, but you have the benefit of the video, and hopefully I'll be able to do all that magic and send you the link by this afternoon. Questions, comments? No? People good?
Do you want us to read chapters 4 and 5 and summary?
Yeah, so finally you can do summary of 3 plus the lecture and just one single summary, all right? You can keep it to 2 pages, that'll be great. If you can't, well, do whatever you can. But don't get too much into the detail, give me the big picture, all right? Okay, excellent. Thank you very much, and we'll see you next Saturday.
Thank you.
Thanks a lot. Excellent. Okay.